I'm Administrator to the OECD Adverse Outcome Pathways Program, and I'm so glad and pleased to welcome you to our second webinar on Adverse Outcome Pathways. So thank you all for joining in and for your interest in this topic, and we really appreciate your time, and we surely hope you find this webinar useful and valuable. As I've already said, this is our second webinar on AOPs. The first one was on general concepts, the objectives and use of AOPs, whereas today's webinar is focused on assembling and evaluating weight of evidence and quantitative information for AOPs. The purpose of today's webinar is to explain why weight of evidence is important for AOP development. We'll also present the types of evidence that are assembled, and we'll share with you some great examples demonstrating the lines of evidence. Finally, we'll talk about quantitative understanding of AOPs. Basically, we'll see what are the quantitative AOPs and why they are developed. So to help me complete these objectives, I'm joined by three experts: Bette Meek from the University of Ottawa, Kristie Sullivan from the Physicians Committee for Responsible Medicine, and Dan Villeneuve from the US Environment Protection Agency. The three of them have been pivotal to helping us drafting the OECD Users Handbook and guidance on AOP development and assessment. And they are also passionate about the AOPs with many years of experience in AOP development, training, and reviewing. They have done a tremendous job supporting the OECD AOP program since its beginning, and we're very happy to have them here with us today. So before we get starting, I would like to share a few housekeeping items. So first, we're recording today's webinar. The recording will be shared with you within few days, and feel free to share it with your colleagues who were not able to be here with us today, and with your networks through the social media. Also, because of the large number of participants, all participants are muted on today's call, and you're requested to stay muted at all time. We encourage you to ask questions by submitting them to the chat box. So we'll have time for questions after the three presentations, and we'll try to answer as many as we can after the call. You can also email us specific questions that we may not have time to answer today, as I'll indicate at the end of the webinar. And with this, I will pass the mic to Bette Meek, who will give us an introduction to weight of evidence for AOPs. Over to you, Bette.
Okay, well, it's a pleasure to be here today to talk to you a little bit about just to introduce the concept of weight of evidence for adverse outcome pathways, to present some kind of concepts associated with weight of evidence, and Kristie will follow up providing more detailed examples. So good morning, afternoon, and evening to all. So by way of a very brief outline, what I'd like to talk about today is what is weight of evidence analysis for adverse outcome pathways, why it's necessary and important, and a little bit about what it entails. What is weight of evidence analysis for adverse outcome pathways? Weight of evidence is actually a comprehensive, transparent judgment concerning the extent of the supporting evidence. For adverse outcome pathways, it's based on clearly defined considerations, which are well documented in the guidance and handbook for adverse outcome pathway development. And those considerations do draw upon considerable previous experience in the assessment of chemicals by regulatory agencies. So why is weight of evidence analysis for adverse outcome pathways necessary and important? Important to facilitate application of adverse outcome pathways for different purposes, as, for example, components of priority setting, screening, and full hazard and risk assessment. Essentially, it helps us to organize data on key events. It helps us, for example, in some of the applications, to assess biological plausibility of associations in epidemiological studies. Adverse outcome pathways are also applied in environmental monitoring and in developing testing strategies. And so we need to have some measure of the confidence that we have in the extent of supporting data for both components of and the entire adverse outcome pathway to understand how best to apply them. And it really constitutes, these considerations of weight of evidence really constitute the bridge between research and regulatory application. You can think of them as a kind of knowledge transfer. It provides opportunity to the research or the development community to communicate knowledge in a format which supports regulatory application, essentially enables them to transfer their much greater knowledge of the biology to the application community, who in this case is the regulatory community. It also provides opportunity to the regulatory community to increase understanding in development of aspects that are relevant to tailor the research for regulatory application. Essentially informs the developer about the nature of the studies that they might conduct to support regulatory application. So what does weight of evidence entail? So it's based on modified Bradford Hill considerations, and many of you may well be aware of the Bradford Hill considerations because they're applied quite broadly by different communities and in different domains. The Bradford Hill considerations were introduced fairly early on in about the mid-1950s to assess causality of associations in epidemiological studies. But they've subsequently been adopted by a wide range of communities, as I'd indicated, for many different applications. For adverse outcome pathway assessment, we've chosen those Bradford Hill considerations that are most relevant for this particular purpose. And we've selected those Bradford Hill considerations based on regulatory experience in assessing chemical-specific mechanistic data, or what we refer to as mode of action analysis. Those considerations continue to evolve with additional experience in assessment and application. So they're very much expert informed and experience based. So if we think about the adverse outcome pathway as being the chemical agnostic biological pathway, or the toxicodynamic portion of the pathway, that really describes key events between the molecular initiating event and the adverse outcome. And both mode of action and adverse outcome pathways are really based on a very simple conceptual construct of dividing this pathway between exposure and effect into a series of key events. When we add in the chemical specific toxicokinetic piece, we're talking about mode of action in that case. So a chemical and the chemical specific aspects will trigger the disease process, and the disease process is really described by the adverse outcome pathway. So there was a framework developed in the in the early 2000s to kind of systematically assess the extent of the evidence for chemical specific mode of action analysis. In 2014, there was an update to that framework to address kind of evolving technology, and also to consider the impact of the experience that we'd had to date on the kind of definition and application of the Bradford Hill considerations. There was another, a follow-up publication to that publication the same year. And so all of that experience has now been incorporated in the user's handbook supplement to the guidance document for adverse outcome pathways associated with the OECD program. So of course, the guidance and handbook are inextricably linked to the adverse outcome pathway knowledge base and its user-friendly interface, the Adverse Outcome Pathway Wiki. So this is the description of adverse outcome pathways that is entered by the developer into the wiki. And there are two parts of these descriptions that address weight of evidence considerations, the key event relationships and associations, and the overall assessment of the adverse outcome pathway. There's a critically important annex in the handbook, which addresses, kind of summarizes all of the considerations that, in fact, provide the basis for our weight of evidence considerations for adverse outcome pathways. So it's called Annex 1, and essentially what it does is describes the considerations that are important, very clearly indicates the defining questions for those considerations, what type of data we're asking the developers to consider, and provides examples of data sets associated with high, moderate, and low confidence. And underlying that annex, there's a number of other templates to essentially which provides guidance to the developer to characterize the supporting data. So there's a lot of guidance in the handbook on how to do adverse outcome pathway kind of weight of evidence analysis. So what are the Bradford Hill considerations we use? The first is biological plausibility of the key event relationships. So essentially that is the extent of knowledge of the biology of the pathway. So how well has that pathway been documented? Do we know enough about it to be able to predict what would happen if we disturb the pathway? And that, of course, forms the basis for ways that we can test our knowledge of the pathway. So, for example, is it well known in human disease, and the pathways actually, our knowledge of the pathway has served as a basis for therapeutic intervention? Or is it based more on just our knowledge of the structural functional relationships where, for example, we know that if a chemical is impacting a liver cell, that in fact, if you impact enough of those cells at a different level of biological organization, that may impact the function of the liver. The second Bradford Hill consideration is, or modified Bradford Hill consideration is essentiality of key events within the adverse outcome pathway. And that essentially addresses the necessity of the key events. So by definition, a key event is necessary prior to the adverse outcome pathway. So prior to the adverse outcome in the description of an adverse outcome pathway. And so we're essentially defining the type of experimental support we're looking for to support the essentiality of the key events within the adverse outcome pathway. It's normally experimental support from specialized studies to block or modify key events and stop recovery studies. So if we stop or block an early key event, what's the impact on the later key events? Is it consistent with what we expect if that key event is essential? And the final consideration is empirical support of the key event relationship. So that's the pattern of quantitative associations across key events. And we often consider that through the application of stressors. So what we would do then is apply stressors and then take a look at the pattern of the quantitative associations, which we can rather predict based on the adverse outcome pathway being the consideration of key events at different levels of biological organization. And some of those considerations that we take into account relate to, for example, is the incidence of the earlier key events greater than it is for the later key events? Do the early key events occur earlier than the later key events? And those considerations we often look at across key events in concordance table, and Kristie will provide an example of one of those. Each of these considerations, we have these example data sets associated with high, moderate, and low confidence. And we rank order the considerations from more important to less important. And again, that draws upon previous experience in assessing mode of action. So the objective then is really to provide a kind of snapshot view, and there's a graphical interface in the Adverse Outcome Pathway Wiki that does this, where we're giving the user, in this case, the regulatory community, some idea of where there are data gaps in the extent of documentation for the adverse outcome pathway and where you have really high confidence. So, for example, we illustrate the confidence that we have in the biological plausibility and empirical support for each of the key event relationships, based on the sizes of the arrows. For the essentiality of KEs, we illustrate that by the size of the nodes. And while we make one call across the adverse outcome pathway, we can still indicate the extent of the supporting data for each of the key events. And you can imagine for the quantitation of the key event relationships, which Dan will speak about in the final presentation, we can also indicate the extent of the data supporting those. And the objective really of those indicators is really to provide guidance to the application community as they're considering applying adverse outcome pathways as a component of screening and prioritization, for example, or informed approaches to testing and assessment, or qualitative or quantitative hazard characterization. And for example, if you were considering an adverse outcome pathway for use in quantitative hazard assessment, you would really want very high confidence on the kind of late key events, particularly in this period as we transition from kind of animal testing to more kind of predictive testing. If you were developing testing strategies, you'd want high confidence in the earlier key events particularly. So the takeaway message on weight of evidence for adverse outcome pathways is it really is about making adverse outcome pathways relevant to application, drawing on regulatory experience, bridging the interface between the research and regulatory communities by focusing or informing on the types of studies that address weight of evidence elements essential for regulatory application, and for providing robust and transparent documentation of the extent of the applications supporting adverse outcome pathways and their components to facilitate application. And I'll leave it at that, and thank you very much for your attention.
Okay, going to share my slide here. Okay. I trust you can see my slides. Hi, everyone. I'm going to be using AOPs entered into the wiki to show how authors have been fulfilling the weight of evidence guidance laid out in the user's handbook, and to highlight what types of evidence lead to different characterizations. So to reinforce Betty's presentation briefly, the three main criteria to consider are the biological plausibility and empirical support of the key event relationships, and the essentiality of the key events within the AOP. On your right are some reference links. The first link is to the user's handbook, which contains detailed guidance on how to outline the weight of evidence in an AOP. And the second link goes to a page on the AOP wiki itself, where some guidance is also provided. For the biological plausibility of the key event relationships, the defining question is there a mechanistic relationship between an upstream and the downstream key event bordering that key event relationship that is consistent with established biological knowledge? Authors can consider the following criteria to determine whether to designate high, medium, or low level of confidence to each key event relationship. And so for a high level of confidence, we're looking for an extensive understanding of the functional or structural relationship based on lots of documentation and really broad acceptance in the scientific community or the literature. For a designation of a moderate level of confidence, it's that the relationship is plausible, maybe based on an analogy to similar biological relationships, but that we don't understand completely the relationship. And a low level of confidence means that there is some empirical support for that statistical association between the two key events, but that there's really no understanding of that relationship. The first example is AOP number 3, inhibition of the mitochondrial complex 1 of nigrostriatal neurons, Parkinsonian motor deficits, by Anna Ball-Price and colleagues. The authors have designated the key event relationship linking key event. I'm not going to go into detail as to the but we'll focus on 2 key event relationships between key event 1 and 2 and key events 3 and 4. And so the authors have designated key event relationship between numbers 1 and 2, inhibition of complex leading to mitochondrial dysfunction, as having high confidence, because there is extensive understanding of this structural and functional relationship between these two events. So it's well known that inhibition of complex 1 leads to failure to produce ATP, increasing production of reactive oxygen species. Also, complex 1 is a main site at which electron leakage to oxygen occurs, resulting in oxidative stress, and that these pathological mechanisms are actually so well studied as to be used as markers for evaluation of mitochondrial dysfunction. And I'll just note here that this description, I've really pulled out the key phrases to present, but I would encourage you to go to the AOP itself to read more text and references in detail, and in fact, take a look at some other AOPs to see the breadth of and get a sense of some of the descriptions. So I'll just use a little pointer here. This diagram from Dauer and Prozborsky indicates this relationship. So we have a clear understanding that within the mitochondria, blocking complex 1 interrupts the transfer of electrons through to ubiquinone, and perturbation of this process decreases synthesis of ATP and increases the production of reactive oxygen species. The key event relationship linking key event number 3 to number 4, impaired proteostasis, leading to degeneration of neurons of the nigrostriatal pathway, was designated as a moderate level of confidence because while there is reasonable biological plausibility by analogy with a similar pathway, there isn't a complete understanding of the mechanism at work leading to cell death and degeneration of the neurons. So again, we turn to this review to depict this situation. So you can see here that it remains unclear whether misfolded proteins or proteostasis directly causes toxicity or damages cells through the formation of Lewy bodies. There's some controversy whether Lewy bodies promote toxicity or indeed lead to neuroprotection by sequestering misfolded proteins. And it's thought that there is some relationship with oxidative stress, ATP depletion, and all of that leading to programmed cell death machinery and then neurodegeneration. So again, there is evidence, but it's not completely understood. For the assessment of essentiality of the key events, we're looking to see an impact on the downstream key event or the adverse outcome if an upstream key event is modified or prevented. If a designation of high confidence is made for essentiality, that often means that there is direct evidence from specifically designed studies demonstrating an impact on the pathway. And so again, blocking, for example, an upstream key event, does that affect the occurrence of a downstream key event? This reinforces the AOP development principle that key events should be necessary for the further progression of the pathway. Moderate level of confidence indicates there's some indirect evidence that the modification of this key event is associated with a change in the downstream key event, and a low level of confidence that there is no evidence or contradictory evidence of the essentiality of any of the key events. Just to note that direct evidence, what we're referring to there is often that there is a clear disturbance of the pathway itself, while indirect evidence, you have a demonstration of disturbance of some factor impacting the pathway. While the consideration of whether each key event is conducted separately, as Bette said, the determination of high, medium, or low confidence is also conducted for the AOP as a whole, based on the extent of available evidence of the range of key events in the AOP. So for essentiality, we're going to consider AOP number 15, alkylation of DNA in male premeiotic germ cells leading to heritable mutation. This is an AOP by Carol Yock and colleagues. The best evidence for essentiality in this AOP is for key event 1, insufficient or incorrect DNA repair. The authors have indicated a moderate support for essentiality of the AOP's key events as a whole. While there is direct evidence for essentiality of key event 1, as outlined in the bullets on your screen here, including both blocking of mutation repair and enhancement, the rest of the AOP does not have appreciable information that would lead to a high call for the entire AOP. This is indicated by the authors in the rest of the discussion, and the main points are that the molecular initiating event, alkylation of DNA, cannot be blocked, and likewise mutations transmitted to offspring cannot be blocked or removed, and so it's difficult to show the essentiality of those key events. Finally, I have an example for the demonstration of an empirical support criterion. First, the question authors should consider is whether an upstream key event occurs at lower doses and earlier time points than a downstream key event, or at the same dose of a chemical or stressor is the incidence of upstream key events greater than for downstream key events. For a high level of confidence, you're looking for multiple studies showing this dependent change in both events, following exposure to a number of specific stressors or chemicals. For a moderate level of confidence, you're looking for, or you may see a demonstrated dependent change in both events following exposure to maybe a smaller number of stressors, or maybe there is some evidence that's inconsistent with an expected pattern. If you're seeing a low level of confidence, you may have limited or no studies reporting this change, or there may be significant inconsistencies. The descriptions put forward by authors for showing empirical support for the key event relationships will vary depending on the AOP and the evidence available to support it. You may see descriptions in table form, bullets, or in paragraphs. Ideally, you would see or will provide data tables showing that dose response and temporal concordance across studies exists. And authors should describe or address consistency across the studies and any inconsistencies or uncertainties. So again, for empirical support, we're looking for toxicological data that demonstrates that an upstream key event occurs at lower doses or earlier time points than a downstream key event. And at the same dose of a chemical, do you have a higher incidence of upstream key event than the downstream key event? We're coming back to AOP number 3 for a discussion of how authors can use this information and demonstrate an answer to this question. In this table, we have a stressor for the AOP, and the authors have provided a high level of confidence to this evidence. Now we can see here's the stressor, and we have different concentrations, and we can see the different key events occurring at earlier and then later time points as you move down the pathway, from 4 hours to 24 to 5 weeks. The plus signs in the table are low to high severity scores. And so you can see as you move to higher concentrations or further down the pathway for this study, the severity for each the key events increases. Now, as you can see, the time concordance from 4 to 72 hours and then 24 hours for the later key event is slightly overlapping. And this is fairly common actually when you're reviewing existing data from studies that weren't necessarily specifically designed with AOP development in mind. And it's likely because of the original experimental design rather than necessarily some deficit in the data, but that's just something to keep in mind. And I will also note that there's much more detailed information for each key event relationship on the AOP and each key event relationship page. So the table doesn't take the place of all of the references and information that you might need, but this table does make it a really easy way to quickly assess the available evidence. So I'd like to just conclude. In this presentation, I've tried to give you a sense of how authors of a variety of AOPs describe the weight of evidence behind the levels of confidence they have designated for various elements in the AOP, and also what to look for if you're trying to evaluate the weight of evidence of any particular AOP. Guidance has been provided to increase the fidelity and consistency of the evidence evaluations in the face of many different kinds of AOPs with a variety of evidence underpinning them. And as more experience is gained within the AOP and the toxicological community, AOP evidence evaluations will continue to be refined in order to ensure descriptions are useful for users and straightforward to implement for authors. Again, to remind you, add your questions to the chat box to have them asked during the Q&A. And now I'm going to turn it over to Dan Villeneuve from US EPA to discuss quantitative evidence.
Between the various key events in the AOP. So what I would like to talk about now is how we develop quantitative understanding of those relationships and why that's important to the application of AOPs. So key event relationships are really the focus, and that's where we capture this quantitative understanding. You'll find it on the key event relationship pages in the AOP wiki, and we really view key event relationships as the workhorse of the AOP. They provide sort of a biological if-then statement. If event A occurs, then we can expect some other event, event B, to happen based on what we understand about the biology, based on evidence from past experiments and past experience and past experimentation. And we also try to define the domain in which that relationship applies. So for what species, sexes, and life stages of organisms does this relationship hold up? So that's the kind of information we're capturing in the key event relationships, and those are the relationships that then allow extrapolation along the AOP. AOPs are described in the AOP wiki, or as we represent them graphically, sort of with the assumption that the magnitude and duration of perturbation or exposure to the stressor is sufficient to drive that pathway all the way to the adverse outcome, that we're basically toppling each domino in the chain or tripping each key event leading to the adverse outcome, and that all adaptive, compensatory, and repair mechanisms have been overwhelmed. And used in that context, in that descriptive sort of way, AOPs are useful for hazard characterization. They say, if we're exposed to a stressor, here's what could happen and why that could happen. Okay? But we know, that's essentially saying that we're standing on a cliff. That's a hazard. We've characterized a hazard. Okay? But for many decisions that we need to make, many regulatory decisions, what's important is not only to know that there's a hazard, that we're standing on a cliff, but understanding what is the actual probability or the likelihood that we're going to fall over the cliff. And so again, for many risk assessment or risk management decisions, it's not enough to know that this is something that could happen if we perturb this particular stressor and why that can happen, but we need to know whether that's actually likely given a specific exposure scenario. So we really need to get at this question of what's the probability that we're going to fall over the cliff, so to speak. So in order to understand that, AOPs really give us this understanding that there is a hazard associated with particular exposure, that we are standing on the edge of the cliff. We need to combine that understanding from the AOP with an understanding of exposure. So, what stressor are we being exposed to? What is the potency of that stressor? What's the duration and the concentration of that exposure? That all plays into whether or not we're going to tip over the cliff. And then there's also considerations of what's our vulnerability as a particular organism or as an individual. What are the factors that influence that vulnerability? And this consideration of vulnerability is some of the considerations that we build into this understanding of the quantitative understanding of these relationships that underlie the AOP. And to get at this question of what is the quantitative relationship between event A and event B, we capture that in the section on quantitative understanding of these key event relationships. And so again, the goal is really to understand what is known about how much change in event A is needed to evoke some amount of change in key event B, and what are the conditions under which we expect that relationship to hold up. And so in trying to capture this type of information, there's really 4 types of considerations or 4 types of information we're trying to gather. The first is defining a response-response relationship. So what type of function, model, or plot describes change in event B as a function of some change in event A. Second, we want to capture information on the time scale. How long after a change in A is event B actually impacted? Are there known modulating factors? So what variables might shift or alter that response-response relationship? And are there known positive or negative feedback loops that are involved, such that what happens to event B can actually feed back to influence what's going on with event A? So we'll go through each of those in a little bit more detail. So many of us in chemical safety assessment are toxicologists, and we're used to thinking about dose response. And so in dose response, we have chemical concentration on our x-axis, and we're looking at the effect on some biological endpoint on our y-axis, some magnitude of effect, typically this sigmoidal type shape, and that's basically a dose response curve. Response is very similar. The only difference here being that rather than having concentration or dose of the stressor on the x-axis, we have some magnitude of effect on event A, such as, say, a % inhibition of an enzyme activity, and then on the y-axis, magnitude of effect on event B, so the downstream event. And essentially, we can plot that just like we would for a concentration-response relationship. We can fit an equation or some function to describe that plot, and that's essentially a response-response relationship. Now, that can take many different shapes. Could be linear, could be sigmoidal, could be U-shaped, or it could, depending on the dynamics involved in this interaction, could actually be a more complex function that we can't really represent in 2 dimensions. We may need to develop something like a model based on a system of ordinary differential equations or something like that to actually represent it. But nonetheless, the goal is to develop some kind of mathematical function that represents how this change in event A translates into some effect on event B. We can see examples of what this looks like from some AOPs in the AOP wiki that actually have some of this response-response relationships described. So this is an example from Adverse Outcome Pathway 25, which links inhibition of the enzyme aromatase to impaired reproduction in fish. Here, describing the key event relationship that links inhibition of aromatase activity to decreases in circulating concentrations of estradiol, an important female hormone. And so you can see from this relationship on the x-axis, we describe the percent inhibition of the aromatase enzyme as measured in something like an in vitro bioassay. And on the y-axis, predicting the circulating plasma concentration of estradiol, in this case in a fish model. And we can fit a regression line to this, and that's essentially what we see down here in this equation, where we could plug in any value for our percent aromatase inhibition and predict y, which is our plasma estradiol concentration. There are other examples of this in the wiki. So here again for AOP 42, linking inhibition of thyroid peroxidase to the development of brain malformations in developing rodent models. We can focus on this key event relationship that links concentrations of thyroid hormone in the serum of the pregnant female to the size of the brain malformation that occurs in her offspring. And so again, here we see key event A, this concentration of thyroid hormone in the serum of the female expressed as a percent of the controls, and a measurement of the volume of that brain malformation that forms in the offspring. And so we can use this linear regression to basically predict this downstream key event based on some measure or prediction of that upstream key event. Again, allowing us to extrapolate down the pathway. Second key component that we're trying to capture relative to quantitative understanding is some indicator of the time scale over which that transition from key event A to key event B happens. This time scale, we know as we go up biological levels of organization, we tend to increase the time scale at which different events are occurring. So at the molecular and cellular level, things happen very quickly on the order of seconds, minutes, maybe hours. As we get up to tissues and organs, we're talking hours, days, maybe weeks for pathologies to develop. And then as we get out to impacts on the individual or even populations, we can often be talking about scales of weeks or months, maybe years. And so understanding the time scale at which this transition from event A to event B happens really impacts an understanding of which assays or measurements are actually practical to perform. If we have a transition that's occurring in fractions of a second, that's maybe too fast for us to actually measure experimentally. And so should we be trying to represent that as key events, or should we expect to be able to measure that particular key event? Likewise, if we're out here where we're looking at scales of weeks, months, or years, is it really practical to make those measurements in an experimental scenario? And do we have to rely on alternative methods like the use of models or the use of epidemiological techniques in order to establish those relationships? And so this understanding of time scale really impacts how you might design experiments to derive these response-response relationships or verify model predictions or predictions based on your AOP. And it really impacts whether modeling is needed, whether the relationship occurs under a time scale that's either too short or too long to practically measure under most experimental conditions. The third type of information we're trying to capture is modulating factors. So what are the variables or other factors that could influence whether an individual or a certain species may be more or less susceptible to a given stressor? This could include, modulating factors could include things like diet, genetic predispositions. Certain genetic populations may be more or less sensitive whether there are other pre-existing disease states that can make one more or less vulnerable to a given stressor, whether there are exposures to other stressors previous to or co-occurring with the stressor exposure in question, or whether there are other environmental stresses that may contribute. And so I think we're all aware of studies in the literature that identify these modulating factors. For example, the influence of a high-fat diet on the development of tumor formation along a particular pathway. And so where we have this information about some of these variables that could actually influence the response-response relationships and make an organism more or less sensitive, we try to capture that information. Now, within AOPs, we don't just want to speculate, but where there's actually evidence and an understanding of how these factors may shift that relationship, where that's understood, we want to capture that kind of information. The final piece of information that we want to capture relative to these quantitative understanding of the key event relationships is really whether there's any positive or negative feedback between event A and event B, such that these aren't operating independent of one another, and they're not just operating where event A impacts event B, but rather event B can have some influence on what's happening with event A. So this is an example, again, from AOP 25, where we have this key event relationship linking inhibition of the enzyme aromatase to circulating concentrations of estradiol. What you see is that as estradiol concentrations fall, there's a feedback response that occurs in the hypothalamus and pituitary, leading to release of gonadotropins and ultimately increases in aromatase gene expression. So as you decrease these plasma estradiol concentrations in event B, you increase the expression of a gene that then can feedback and actually increase the production of the aromatase enzyme, offsetting or compensating for that initial inhibition. And so in order to understand what that response-response relationship is and what the dose-response time course behaviors associated with that are, you have to understand this feedback that occurs between event B and event A. And there are many other examples of this. In some cases where it's event B directly interacting with some part of the pathway to feedback. In other cases, it may involve biology that's not explicitly captured in the key events, but may still allow for interactions with other AOPs or other key events that may then influence this relationship between A and B. So again, we want to capture that information wherever possible. So in summary, in terms of quantitative understanding of the key event relationships, we're trying to capture information on response-response relationships, the time scale of the transition between event A and event B, any modulating factors that can shift or alter that response-response relationship, and feedback mechanisms that could both influence the shape or the nature of that response-response relationship and potentially lead to more complex interactions with other AOPs. As we put this information together for the key event relationships along an AOP, collectively we can link those together to construct a quantitative AOP. And a quantitative AOP then is really an AOP that has sufficient understanding of quantitative understanding of the relationships to allow for quantitative prediction of the probability or severity of the adverse outcome occurring given a specific level of activation or perturbation of the molecular initiating event or one of the early key events in the pathway. And so essentially, where we have this quantitative understanding, we no longer need to assume that the tipping points are occurring, but whether we can evaluate for a given exposure scenario whether we're likely to exceed these tipping points and actually fall over the cliff. So in essence, coupled with exposure considerations, It's the quantitative understanding of the relationships that underlie the AOP that really facilitate the use of AOPs to support not just hazard characterization, but also risk assessment. So with that, I'd like to again remind you to, if you have questions, please type them in the chat box, and we'll be having some discussion of questions shortly. I'd also like to just take the opportunity to announce to everyone on the webinar that EPA is launching a prize competition. The prize competition is called the Target Ecotox Challenge. EPA is offering a $300,000 US prize to company, organization, or team that can provide high quality, low cost technologies or platforms for evaluating global gene expression in samples from 4 commonly used aquatic toxicity test organisms. And these platforms are intended to support the development of high-throughput transcriptomics assays for use in high-throughput screening programs to screen for potential ecotoxicological effects. And so if you're interested or you know of any individuals or companies who might be interested in taking on this challenge, I would encourage you to go to the website shown here. And with that brief side, I will turn the screen back over to Magda.
Okay, great. Thank you so much. Well, it was absolutely fantastic. And I would like to thank at this point our 3 presenters for doing a great job. Like I said in the beginning, they've been great, helping us a lot from the beginning of the AOP program. So we've heard great information that I hope it will help you in the future. So before we move to the Q&A session, and please feel free to submit questions, just to conclude based on what we've heard so far. So weight of evidence evaluation provides transparency and confidence on assembling scientific knowledge in AOPs. Quantitative AOPs further enhance the confidence of their use by regulators. And as in our previous webinar, we try to engage more and more scientists in the AOP development. And I hope you found these 3 presentations inspiring so you can visit the AOP knowledge base and contribute to AOP development. I think that data generated in your lab can be an important line of evidence that can really strengthen the development of and use of some specific AOPs. And maybe this is the right moment to advertise our next webinar on AOP knowledge base that is scheduled in a couple of weeks from now on the 30th of January. If you haven't registered yet, please remember to do so after the end of this call. So, okay, seeing that we have a few minutes for questions. Okay, so I have a question here, the first that came across. If there are key events that have a very short time scale or that are not possible to measure, how should this be represented in relation to the evidence? Should be then omitted or not? Yeah, I think, Dan, can you take this question?
Sure. So for events that happen on a very short time scale such that they may be impractical to measure on a routine basis, they may still be used as key events and represented as key events in an AOP because they help establish the biological plausibility of a particular mechanism. It may help establish why, for example, inhibition of a particular enzyme or the interaction with a neurotransmitter receptor, why that actually links to some of the downstream biology. And so it may happen on a time scale that's impractical to measure, but there is understanding of the biology involved, and so we might include it as a key event. And in many cases, those measurements may have been made, but can only be made under very specialized experimental conditions that you wouldn't routinely do in sort of a toxicity testing scenario, for example. So we could still capture that information from sort of the basic biology research areas that we might not measure routinely in a toxicity type study. Likewise, if there's that biological plausibility to support the weight of evidence, we still might capture that. But in general, we tend to want to focus on key events that we can use to verify that the biology is responding as expected when exposed to a stressor. So often we would try not to necessarily represent a lot of key events that we can't practically measure. Hope that answers the question.
Okay, thank you, Dan. I don't know if the other two presenters want to anything to add or to elaborate on, or we move to the next question. Yeah, go ahead.
Magda, it's Bette. Just to elaborate slightly on that, I think in general, as Dan's indicated, we kind of focus on those key events that we can measure because that's likely where we're going to have most of the evidence as well as support from a weight of evidence perspective.
Okay, and we have one more question about the role of epidemiology in AOP, how we capture epidemiological data in AOPs.
Yeah, certainly. Although I think probably Bette would be better suited. But what's nice about the AOP framework is that it's intended to capture lots of different lines of evidence and allow one to kind of see them all in one place and view the linkages and compare them with each other. So just like toxicological data, an AOP author would include any epidemiological data that's available and judge it within the guidance of that's provided for weight of evidence for the various elements of the AOP. So, is the evidence consistent in terms of time scale, in terms of different levels of exposure? Is the biological relationship consistent? Ideally, if you have looking at biological plausibility, you may have different lines of evidence and they're all consistent with each other. That's a good sign. So that's kind of what we're looking for with biological plausibility. So the role of epidemiology information is just to include in the pathway as a line of evidence. I don't know if Bette wants to expand on that.
Just a little more. No, I think you've covered it, Kristie. I think that obviously we're talking about normally we're measuring quite late key events in epidemiological studies. So you're talking about the far end of the adverse outcome pathway, but also certainly we get quantitative relationships from epidemiological studies as well. So they also contribute to quantitation of the key event relations, and normally for the late events in the adverse outcome pathway.
And to conclude, here is the last question. So if a key event has two outcomes that are both plausible, okay, and that the pathway can go back to this point and again go two ways, but these outcomes are different, how should this be represented? A split in the pathway or two different AOPs?
Hi, this is Dan. So generally, if a given stressor or a given molecular initiating event or key event could lead to two different outcomes, those tend to be represented as two different AOPs, but because they share key events along the pathway, they would also be represented as branches within an AOP network. And so, part of the way AOPs are represented and the way they're represented in the AOP wiki in terms of separate pages for each key event and each key event relationship, that those pages can be shared among multiple AOPs, and essentially that forms a network. And so you could, by taking two AOPs, combining them together, looking at the shared key events, you could then understand where those branch points are within the network. And you may see that one of those outcomes may happen in a certain target tissue and the other outcome may be in a different tissue, or one of those outcomes may occur in certain species but not in others. Some of those outcomes may be occurring simultaneously in the same organism that's exposed. And so you can use that view of the AOP network to see what these multiple potential outcomes are to the same stressor or the same activation of the same event upstream.
Okay, so seeing that, okay, we past the an hour of webinar, I suggest that for some of the other questions, basically, you should visit our website, recordings of past webinars and training material. And hopefully we'll try to contact you with the replies to the questions that we were not able to answer now. So with that, I think that we are close to the end of the webinar, and I will wrap us up here. But again, I would like to thank our three presenters for speaking and sharing their time and expertise today, and of course all of you for attending. To find us in social media, and if you have any hanging questions, please don't hesitate to email us. This is Natalie Delroux and Magda Sachana. I appreciate everybody's time, and I hope you enjoy the rest of your day, and hopefully see you soon in our next webinar on the 30th of January. Thank you very much. Bye, everyone.